This is lecture 13 material on DNA transformation. And you'll remember that we talked about this earlier in this semester because there were experiments done by Fred Griffiths and later Avery, McLeod, and McCarty that led to the identification of DNA as the hereditary material because of their experiments with bacterial transformation. Now, very briefly, this process involves DNA, naked DNA, that gets taken up by a cell, and in the process of perhaps integrating some of that new DNA that's taken up into the recipient cell, they become genetically transformed. And we'll talk about differences in gram-positive and gram-negative organisms. Very briefly, we'll talk about artificial transformation because not all bacterial cells can be competent. In other words, not all bacterial cells will be able to take up DNA. And then finally, we'll look at the regulation of competence, and we'll focus specifically on a process called quorum sensing. So the significance of this process is that in some organisms, taking up DNA is useful because DNA can be used as a food source. That doesn't happen in all organisms, but DNA does serve an important role in repair of damaged chromosomes, for example, in organisms that are competent. And finally, this contributes a lot to genetic diversity and the evolution of variation in different organisms. Now, I'll focus most of this lecture on cells that are naturally competent, but I want to remind you that not all bacterial cells can be competent and take up DNA, so there are processes that people have developed to artificially induce competence in organisms such as E. coli. I want to talk about some experimental approaches that were used to study transformation. So in all these experiments, they're mixing DNA from a wild type that's Rg plus, and they're mixing with an Rg minus recipient, so that's what is shown here. And of course what they'll be measuring is if DNA gets in and it recombines, we'll get Rg plus out of this, some prototrophs. So to measure how fast it takes, how long it takes for the DNA to get in and recombine, they do the following experiment. They mix the DNA with the Rg minus mutant. They allow it to incubate for a certain time, and initially they do this for a very short period of time, and then they add DNase. DNase is going to degrade any DNA that has not gotten into the cell. Now, in order to assay what actually got into the cell, they do the next set of experiments with this. They now take the DNA from the cells, and remember there's nothing outside the cell, we've already degraded that. They isolate that DNA and they now add it to the outside of a new set of cells. They're also Rg minus, and they do the transformation experiment a second time. Now, when they initially do this and they have added DNase very quickly, they get no Rg minus transformants. But they redo the experiment a second time, and in this case, they add DNA again, and they, as usual, add DNase, but they have let the reaction go a little bit longer. So this time, some of the DNA gets in, and notice that we start with double-stranded DNA, but what gets into the cell is single-stranded DNA. But now they stop that reaction again, and they isolate. They've added DNA, so we don't have anything outside anymore, and now they extract the DNA from inside, and they now again add it to a fresh Rg minus recipient. In this case, they still get no Rg plus transformants, and that's because in this case the DNA got into the cell, but it was still single-stranded up here. Go back up to the cell that they used as their marker. It was still single-stranded DNA and it hadn't recombined. So when they've isolated DNA from this cell and they put it down here, what they really have is a bunch of double-stranded DNA that was from the chromosome and all this single-stranded DNA, but that is insufficient to get in and give us Rg plus transformants. So the very last experiment that they do, they again allow time before adding DNase, and this time all of the DNA can get into the cell. And you'll notice that it gets in and it's going to be converted to single-stranded DNA on the way in, but in this case it also recombines onto the chromosome. So now when they isolate DNA from these cells after the DNase treatment, they now have double-stranded DNA, and some of that double-stranded DNA carries the ARG plus gene. It will get into the cell, it will become single-stranded, and it will recombine in this case, and now you can get ARG plus transformants. So what they realized from this is that there are really two components that need to be taken into account. One is time. You need time for the double-stranded DNA to get into the cell and to recombine. And number two, they knew that you needed double-stranded DNA as the initial substrate. Overall, the process of transformation is very similar in gram-positive and gram-negative organisms. In both cases, there will be an initial binding that occurs to double-stranded DNA. In a gram-positive, there will be fragmentation of that DNA, and finally transport of a single strand of DNA into the cell. The other strand is degraded and left outside the cell. In a gram-negative cell, after binding, initially the DNA will be in the periplasmic space, and then the machinery on the inner membrane of the cell is actually involved in the transport and degradation process. So minor differences. Here's a figure from your book that shows, again, a comparison between gram-positive and gram-negatives. Let's look first at gram-positives. Again, they only have one membrane, the cytoplasmic membrane, but they do have a thick cell wall, so we need to have these pillin-like proteins out here. COMG is one of the main components of that. COMEA is a double-stranded DNA binding protein, and there's a protein in the membrane, COMEC, and the double-stranded DNA shown here will enter after being recognized by COMEA. A nuclease, very important, will degrade one strand of DNA, and then the other strand of DNA will be taken into the cell. And again, it's very similar in a gram-negative organism. The main difference is that in the outer membrane we see a protein called PIL-Q, which is a secretin. Again, we see a pilus-like structure. In this case, it's made up of a protein called PIL-E, and the inner membrane protein is called COM-A. But again, double-stranded DNA enters in. Again, it's in the periplasmic space initially, where the nuclease will degrade one strand of DNA and single-stranded DNA gets taken into the cell. Here's a little bit more detail, a picture from a recent review article. I want to remind you that even though these are subunits that are nearly identical to a pilus, in this situation it's called a pseudopilus because it's not acting in the same way that a typical pilus does. Remember that pillin subunits are proproteins, so this is just showing the COMG proteins that are going to be processed before they're inserted into this growing pilus. There are a number of proteins such as COMGA and COMFA that contribute to the lengthening of this pseudopilus. Here's COMEA, the protein that I mentioned on the previous slide that is involved in recognizing double-stranded DNA. And here is the COMEC in the membrane of a gram-positive cell that actually brings that single-stranded DNA into the cell. And once inside the cell, there are going to be single-stranded DNA binding proteins that bind that DNA and protect it from degradation. I just want to point out that many of these components that are involved in competence, and by the way, they're typically called COM for competence, COM, There are a number of operons that encode different components. So the COM-E operon, for example, is comprised of 3 different genes. One is for COM-EA, the protein that binds the double-stranded DNA. We don't need to pay too much attention to COM-EB here, but the other one that I've already mentioned to you is COM-EC, which forms this channel in the membrane. We will come back at the end to talking about regulation of this process. So cells don't produce all of these proteins all the time. They do it when the situation and the timing is right. So there is a lot of regulation. And for example, there are proteins such as COM-A, which will be involved in regulating this entire process. I mentioned that the whole process of competence is a highly regulated process in most organisms. And one of the ways that cells do this is by monitoring the production of small molecules called inducers, autoinducers. And in this case, for a gram-positive organism, there's something called a competent stimulating peptide, CSP, that many streptococci, for example, use to monitor and regulate not only competence, but the production of bacteriocins and biofilm formation in these organisms. So remember, many of these are organisms that are going to be causing infections, or they're living in your mouth and they're making biofilms on your teeth. So the regulation is to monitor cell density. So they wait until they're at a high cell density, and that means that a lot of cells are producing this CSP, so it's building up in the environment. And when it builds up, this CSP can bind to a protein, one of the COM proteins. In some streptococci, it's called COM-D, but we'll just say it's a COM protein in the membrane, and that stimulates the production of all of these proteins that we just talked about, such as the pseudopilus, that are going to be involved in DNA uptake. So the consequence of this CSP then is that we get all the pseudopilin, etc. So we get uptake of binding of the double-stranded DNA. The nuclease that degrades one strand takes the DNA in, and we get recombination down here. Well, let's look at another outcome of production of these CSPs. And by the way, these CSPs are typically about 17 amino acid peptides. They're essentially pheromones because they're very potent at very low concentrations in the cell. So here's an example where a CSP can actually result in fratricide, killing of other cells. So this happens when we've got 2 different strains, for example, in this case of Streptococcus pneumoniae, that are in a population, and they have different types of CSPs. So many of these CSPs, these quorum sensing molecules, are produced in response to some environmental signal. In many cases, it's nutrient down regulation, so there's a decrease in nutrients. Now what happens is, in this case, this particular cell that has the gene for CSP1 will start to make CSP1. That will induce all the cells that carry that CSP1 system to become competent so they can take up DNA. But it has an opposite effect on the cells that do not carry that CSP1 gene. They will not become competent, and instead the cells that became competent, as I said, they also start to produce bacteriocins. They produce something called a pneumolysin, and here are some of the lytic enzymes that they produce. They basically start to kill all of the cells in the population that are not like them. So that's why it's called fratricide. So we get down here and we see that our competent cell is now stimulating the lysis of the CSP2 cells. We get what's called allolysis, and the result of that for the population of cells that were competent up here is that the CSP2 cells now lyse, they release their DNA, and that can be taken up by these cells, so that it's a way to perpetuate the diversity of the CSP1 cells in the species. This is a clever experiment that was done in Michael Sheets's lab at Columbia, and it was used to actually determine what the force was that was required to get DNA into the cell. And they did it by doing kind of an experiment in reverse of what would naturally occur. In this case, they took DNA and they bound it in this picture to a ball, and then they mixed this in an optical trap system with Bacillus subtilis cells. And then they actually measured, and you can see this down here in panel B. So here's our optical tweezers right here, and you can see it in each of these figures. It's being held stationary by the optical tweezers, but it's got DNA bound to it. And you can't see the DNA in this figure, but you can certainly see the cell. And what you notice is that this cell at time 0 is pretty far away from our optical trap, but it starts to move in the direction of that optical trap because it's got a molecule of DNA that it is basically taking into the cell, and that is dragging the bacterial cell toward the optical trap. And they can use this to measure the force that is being generated, and they found that it's about 0.04 piconewtons per nanometer, which is pretty remarkable for a bacterial cell. And they did this, they measured this for N was 250 different episodes of this occurrence. Now, when they did this experiment, they not only looked at a wild-type cell, but they also looked at some mutants. And in particular, they looked at strains that had the COM-EC gene removed, so they would make no COM-EC protein. And they also looked at mutants that lacked COM-G, meaning they would not be able to make the pseudopilus. And what's interesting is that you can see that here's our wild-type situation, and you can measure relative binding frequency. It's very high. And in the COM-EC mutant, very surprisingly, there was a very minor change. So that tells us that COM-EC is not absolutely essential for this DNA transport. However, what is essential is COM-G. Very low amount of DNA gets taken into the cell if you lack the pseudopilus. And then finally, here's a control, and if they have no DNA, they see no movement of bacillus toward that optical trap. Let's turn to looking at natural transformation in gram-negative bacteria. Remember, we're talking about organisms such as Haemophilus and Neisseria. E. coli and many of its relatives are not going to be competent to take up DNA. So typically we're looking at some of the components that we've already seen, plus, again, a type 2 secretion system, which is going to be needed for that type 4 pili that we've already looked at, and secretin, which is the component in the outer membrane. And that's really sort of the major difference that we're looking at here. There's another important factor, and that is that some of these organisms, and I'm giving you the examples here of Haemophilus and Neisseria because these are the ones that we most commonly look at for natural transformation. They actually require a recognition sequence. So if DNA is added to these cells that lacks these recognition sequences in the DNA, the DNA will not be taken up by these cells. I want to very briefly describe two more proteins that are unique to the gram-negative organisms in transformation, and one is COM-E. So this is looking at over here a figure from a model from Neisseria, and they were actually comparing the pilus assembly structure. Pili in Neisseria can be used for motility, but you'll see that the competence components are also very, very similar to the type 4 pilus that you see in Neisseria. But now we'll just focus on the pilus component. And again, here's the double-stranded DNA that's being brought in. This barrel structure out here is pill Q, the secretin. And then what they wanted to learn more about was an essential component called COM-E. They knew it was a paraplasmic protein, and they knew that mutations in COM-E blocked the transformation process. So here was a study that was done to work out what COM-E was doing. So here's what they did to test their hypothesis that COM-E was important to actually bind to the DNA that was entering the cell. What they did was they used an expression technology vector to express and purify COM-E. So this is just showing you plasmid controls, for example, that didn't have their gene in it, and then they could make and induce expression of COM-E. They added IPTG, as you'll see here, and then they purified a lot of COM-E protein. And then what's shown in B is that they incubated increasing amounts, that's shown by this black bar here, increasing amounts of COM-E protein, starting with 0, with DNA from Neisseria. So we've got double-stranded DNA, and in this case it's radioactively labeled. We have the DNA on its own with no COM-E migrating at this position in an electrophoretic gel. When we start adding the COM-E protein, we see the DNA is actually retarded in the gel. This is called a gel shift assay, and it shows that we have specific binding of the DNA with the COM-E protein, and that's what slows up the DNA in its migration through the gel. The other protein is pill Q, the secretin. And pill Q is interesting because mutations in pill Q prevent the uptake of DNA, and you can actually add cells that have pill Q that make it, and they'll share pill Q by passing pill Q from one cell to another, and they'll be able to share with a strain that doesn't make it, so that a pill Q mutant can actually be made to uptake DNA. And here's a diagram here that's just showing the DNA is being taken in through this secretin, which forms this large channel in the outer membrane, and the DNA gets into the cell, again, as we talked about earlier, one strand being degraded and single-stranded DNA getting into the cell. Finally, in gram-negatives, I want to also point out that transformation in many organisms, similar to what I mentioned with Streptococcus, is that it requires a nutritional downshift. That means that this process is induced when there's some limiting nutrient. The other unusual feature about Haemophilus, in addition to the fact that it requires that specific recognition sequence in the DNA so that it will only take up certain types of DNA, is that there's an unusual complex that forms. In this case it's called the transformosome, and the DNA actually gets taken up into that transformosome, and then it invaginates into the cell. And what they've found is that when DNA is in this complex, it's actually protected from nuclease degradation. And then, as we've seen before, DNA will get into the cell from the transformosome, and it will be able to recombine. So here's the single strand of DNA on the chromosome, and now daughter cells could potentially give rise to two different phenotypes, because this new incoming DNA could have a mutation that's not existing on the other strand of DNA. So you end up with genetic diversity in this way. Work in Rosie Redfield's lab at University of British Columbia has shown that in this nutritional downshift that occurs, Haemophilus is actually using some of the DNA as a nutrient to prevent it from entering starvation, and some of the DNA, as we've said earlier, ends up being used as a substrate for recombination. Of course, not all organisms can be induced to be competent and take up DNA, so organisms such as E. coli, as we've already talked about, have to be artificially induced to take up DNA. In the past, the traditional way to do this was to treat the cells with calcium chloride and follow that with a heat shock. And that would, first of all, in the calcium chloride treatment, that would form a complex, a DNA complex. And then in the second part, that rise in temperature would allow the DNA to be taken into the cell. That process is a little bit tedious, and it has been superseded by electroporation, which is a pretty fast, easy way to treat cells because you simply wash them and use an electric current in a high capacitor to get DNA into the cells, because it temporarily, this charge will depolarize the membrane and allow the DNA to enter. And then in other microbes such as yeast, eukaryotic cells, polyethylene glycol or lithium acetate treatment has been used to get DNA into cells. As I mentioned earlier, this is a highly regulated process because the cells typically don't want to produce all of the components to take up DNA unless there's an urgent need to do so, for example. Regulation may depend on, as I mentioned earlier, either nutritional downshift or, in some cases that we're going to talk about now, a process called quorum sensing, which measures cell density. And I want to point out also that these are not mutually exclusive. In some cases, a nutritional downshift stimulates this quorum sensing process. So I want to talk a little bit now about the process of quorum sensing and how cells know that they're at a high cell density. So that's what quorum sensing really is. It's a way for cells to measure how many of their like cells are in the vicinity. So here's a very simple diagram that shows the process. And this is a diagram that was based on work that was initially done in Vibrio. And it was actually done by a postdoc in Woody Hastings' lab at Harvard at the time. This was probably almost 40 years ago. And they were working with Vibrio, which is an organism that produces bioluminescence in the ocean. And they found that at low cell density, the cells did not produce light. So they were dark, but when the cells reached a high cell density, they were able to produce light. And they were trying to understand how this light production was regulated, and they did a very elegant experiment. They grew cells to a high cell density when light was being produced, and then they took this sample and they centrifuged it. The cells were in the pellet, and they took the supernate and they added that to cells that were at a low cell density, and they saw that they could stimulate light production. So they reasoned that there was a molecule that was being produced and released into the medium, into the supernatant. So they went about purifying that molecule, and they ended up identifying something that has kind of generically been called homoserine lactones, or you'll also see them written, and I think I will refer to them for the rest of this talk as acyl homoserine lactone, AHLs. So here's again what happens. At low cell density, they are producing this AHL molecule. This is the red circle shown here. But it doesn't build up to a very high concentration because there are not very many cells producing it. However, if the cell density is high, all the cells are producing this, so we get a very high production of this AHL molecule. And when that happens, we get to a density such that the molecule can actually enter back into the cell at a sufficiently high concentration that it binds to a protein called LuxR. And Lux stands for light production, luciferase. And when it binds to that molecule, it will stimulate transcription of genes that will make this enzyme, and we'll get production of light. Now, since those early experiments that were done by Pete Greenberg and Woody Hastings lab. And by the way, Pete Greenberg now works at University of Washington, and he actually studies the production of these quorum sensing molecules in Pseudomonas aeruginosa, and he's specifically interested in cystic fibrosis and the role that these autoinducers play in infection in CF patients. But what has been discovered is that quorum sensing occurs in many, many organisms, not just Vibrio, which is the organism that I just mentioned. It's in a number of different strains of Vibrio. It's in Pseudomonas. It's also, these molecules are also made in different gram-positive organisms. But we can put them into two classes. So autoinducers, as they're called, that's what these small molecules are called. In gram-negatives, they are AHLs for acyl homoserine lactones. And at the core, all of them look like this. They have this lactone ring, and they have an acyl group that can vary. The R group can be anywhere from a small moiety, as in Vibrio harveyi, or a very simple one, as in Pseudomonas, to something that's very complex. You do not see AHLs in gram-positive organisms. However, they do have autoinducers, these molecules that regulate quorum sensing, and in this case they are all these small peptides. So we've referred to some of these before as pheromones in some cases, or you'll simply see them referred to as these autoinducer peptides. For example, here's a short peptide that is found in Bacillus subtilis, and here's a longer peptide that is very specific for Streptococcus pneumoniae. Now that we've established that there are two types of autoinducers, depending on whether or not you are a gram-positive or a gram-negative organism, a peptide in a gram-positive and an AHL in a gram-negative, I want to also let you know that the autoinducer can be interpreted by the cell in two different ways. In some cases, it is internalized, and that was the example with Lux that I talked about, where the autoinducer came into the cell and bound to the LuxR protein. We'll also look at an example later where the autoinducer actually interacts with a receptor protein on the surface of the cell and activates a cascade of events inside the cell. Well, let me review this once again, how quorum sensing works to measure cell density. The example here is for AHL. For a gram-negative, we're looking at a low cell density. We've got a few cells out here. Gram-negatives have a gene called the I gene. It's generically called the I gene. For example, it might be LuxR, but in a different organism it will be given a different name. They make an I protein. The I gene makes an I protein, and that's the protein that makes the AHL. So it's being produced at all times, but in this case we're at a low cell density, so there isn't a lot of AHL. Now we look at a higher cell density after several hours of incubation, for example. We have many more cells, so we have a lot more of the AHL that's being produced, and there's enough of it that can actually diffuse back into the cell. When that happens, it can bind to the R protein, the product of the R gene, the regulator gene. When it binds to the R protein, it activates the R protein, and the R protein, when activated, can bind to DNA and activate transcription of, for example, the light production genes in Vibrio, or if we're looking at a pathogen, it would activate production of virulence genes to make virulence proteins. Well, we've been talking about why quorum sensing is significant in the uptake of DNA to make cells such as Bacillus, Streptococcus, and Neisseria competent, but it's also important in a number of other processes. For example, it's important in symbiosis. Symbiosis being an interaction that occurs between two different organisms to benefit both of them. Vibrio, for example, can form a symbiotic relationship with Euprimnoscolopes, the small squid that's shown in the image here. And Euprimnoscolopes has a light organ, and it needs Vibrio because Vibrio is the light producer in that light organ. And it needs the Vibrio to be in the light organ so that Euprimnoscolopes can develop normally as it's growing. And it also needs to maintain it throughout its life so that it can use light production to protect it from its predators. Quorum sensing is also important in nodulation, and here's an image showing the root hair and the nodule that gets produced. And here's bradyrhizobium moving down an infection thread in this image into the nodule where nitrogen fixation will be occurring. And then a final example is to remind you that quorum sensing plays a very important role in regulating virulence determinants. Two examples, of course, that I've already mentioned, Pseudomonas and Staphylococcus. And the example shown here is an image for Staphylococcus. So here is a gene that is encoding AGRD, and that's going to be processed to give us this small peptide. So this is the quorum sensing molecule, the autoinducer, because this is a gram-positive organism. And in this case, this is an example where the autoinducer will bind to a receptor, AGRC, and activate the expression of, or the activation of other proteins, including AGRA, so that we now activate the expression of a number of genes for virulence components in this organism. This is a final example just looking at how quorum sensing plays a role in regulating competence in bacillus. So here's a bacillus colony over here, and I'll remind you that it's a gram-positive organism, so in this case quorum sensing is going to involve a peptide. So here's the peptide. It's initially produced as a protein, so that means it's going to be processed. A protein called COMQ, and again COM, remember COM stands for competence, so these are all going to play a role in competence, will be involved in modifying COMX, and it gets exported out of the cell, and in that process it's going to have the leader peptide removed, and we end up with our mature protein here. And here is that peptide. And again, it's being also referred to as a pheromone because it's a peptide that's effective at pretty low concentrations. Now that peptide, when it binds a surface receptor, remember I said sometimes they are internalized and sometimes they simply interact with a surface receptor. In this case, the receptor is COMP, a histidine kinase. Kinases are proteins that bind ATP and remove a phosphate. So when binding between the peptide and COMP occurs, we get what's called autophosphorylation of COMP. COMP, even though it stays in the membrane, it can now transfer that phosphate to COMA inside the cell. So this is like a way to signal from the outside to the inside of the cell what's going on outside in the environment. When the phosphate is transferred from COMP to COMA, that converts COMA into an active form. It's like flipping a light switch to turn the light on. When COMA is active, COMA is called a response regulator. And I've run out of room. And it will bind to the chromosome at particular sites, and it will activate transcription. In this case, it's going to activate all those other COM genes that we saw earlier that will allow for the uptake of binding and uptake and processing of DNA.